Chapter 21, Viruses. As we begin our investigations of living things, it is a bit ironic that we're starting off with structures which are not technically alive. So viruses are pathogens, but they do not fall into the category of living things. They do, however, have a large impact on cells. They are not themselves made of cells, but they do impact cells because they require actual cells in order to reproduce and make more. So viruses are acellular. Remember, you put an A in front of word, it means not. So they are not made of cells, and that is because cells have the requirement of having a plasma membrane, cytoplasm, DNA, and ribosomes. And viruses do not have all of these components. They do not have any ability to metabolize, and they can't grow. They rely on their host cells to make copies of themselves in order to reproduce. They're considered a parasite because they can't replicate unless they are using the machinery of the host cell, the ribosomes, etc. Where viruses came from is really not quite understood at this point. They are made of pieces of nucleic acids, so either DNA or RNA from various sources to come together. So they have a variety of nucleic acid makeup. And they can infect nearly all kinds of living cells. So there are viruses specific to bacteria. There are viruses specific to plants. And of course, there are viruses specific to animals. And they are not only species specific, but often tissue specific. So that means that they will infect only particular tissue. For example, the HIV virus only affects T cells. Now I put up here something that always gives me a strong reaction, and this is when I see containers of disinfecting materials like this Clorox, where they say kills cold and flu viruses. Well, keep in mind that you can't really kill a virus because it's not technically alive. You can break it apart and deactivate it, but you can't really kill it. So how long have we known about viruses? Well, we've known that there's been some kind of pathogen which has impacted various living things. Like here we see the tobacco mosaic virus. Here is the virus itself, and this is the result on tobacco plants. So we've known that it's been around because we could filter out the bacteria, and yet the plants would still become infected. We knew something was there, but we weren't able to actually see it like we see it here in this micrograph. So this was back in the late 1800s. So it took new technology to be able to actually see viruses using microscopy, and that's using the electron microscope. And here we have a bacteriophage. So this is a virus, this thing that looks like a little lunar landing module. So this is attacking a bacterial cell. So this is a bacteria cell. This is the virus. And the reason why we can see that is that with electron microscope is that almost all viruses are too tiny to be seen with regular light microscopes like we use in lab. A single virus particle is called a virion, and it's only about 20 nanometers. 20 nanometers. So remember, if you take a meter, you can divide it into millimeters. That's 1,000 of those in 1 meter. You can take 1 millimeter and divide it into micrometers. There's 1,000 of those in 1 millimeter. And then you can take 1 micrometer or micron and divide that into 1,000 and you get nanometers. So these are really, really tiny. Now electron microscopes, which use electron beams for the energy to produce the image, can magnify up to 500,000 times. Remember, our light microscopes can only magnify up to 1,000 times at the max. And the clarity of the image is very good due to a resolution factor of 0.05 nanometers, meaning it can distinguish two different items all the way down to if they are within 0.05 nanometers of each other. Now, the invention of the electron microscopes is what led us to discover various types of viruses that are present. So here we go back to that original picture that we saw. Here is the bacteriophage on a single prokaryotic cell, bacterial cell. And then we look here, their scale is in nanometers. We then scale up. Here is a single prokaryotic cell, so that's one of these, as compared to a eukaryotic cell, and you can really see the huge difference in size. So this little guy would be a tiny, tiny dot on this prokaryotic cell that you couldn't even really see. So virus evolution, how did these viruses come about? Well, as I mentioned before, we're really not completely sure. Did they come somehow from free living cells? Did some bit of nucleic acid escape from a cell and somehow get a coat around it, a protein coat? Did they begin as self-replicating molecules and then lost the ability to do that replication? And we really still are not sure. Let's take a look at what a virus looks like. Viruses are incredibly simplistic. We have a bacteria virus over here, and then we have a couple of animal viruses over here to see. Each virion, which is one virus particle, must have, at a minimum, a nucleic acid. So that could be either DNA or RNA, could be double-stranded or single-stranded, and must have a protein capsid or coat. So if we take a look at our bacteriophage, this is the head part, this is the capsid or the protein coat, and the DNA is inside. Now, if we look at some of our animal viruses, they might look a little bit different, but here's the capsid on an adenovirus, which can cause the common cold, DNA inside, and then we have a RNA virus. So here is the RNA, which is inside of its capsid, which is its protein coat. Now, in addition, some viruses might have what's called an envelope. So that's similar to a cell membrane in that it contains lipids and proteins. It's not exactly the same composition as a cell or a plasma membrane, but this virus, the HIV virus, does have an envelope on the outside. So we see that. The other two do not. So if we look closely at the bacteriophage, as I said, it kind of looks like a little lunar module, landing module. So it has, these are virus particles that infect bacteria. They affect the specific bacteria, Escherichia coli, or E. coli as we know it. So only this particular bacteria. It has DNA for its genome, and we have studied it rather extensively. We know a lot about this bacterial virus. Here's that adenovirus with its capsid coat. It is non-enveloped. It attacks the human respiratory tract. Other viruses which are non-enveloped are things like the polio virus, the papilloma virus, and the hepatitis A virus. And the HIV, this is the one that causes AIDS. So AIDS is the sickness that results from this virus. It is enveloped, so it has that special envelope outside of its capsid. And this is also interesting because it's called a retrovirus. Retro means to go back, and this is because it's an RNA virus, which then has to utilize an RNA polymerase to make it into a DNA form once it gets into its host. So, viruses are made up of nucleic acids. They have a genome. It can be either DNA or RNA, but not both. Maybe single-stranded or double-stranded. A single-stranded RNA or a double-stranded DNA. Maybe circular or linear, so it could be in a circle form or straight line form. And it can also be in one piece or maybe in several pieces. Now, this genome is very, very small. This collection of DNA or RNA is very much smaller than you would find in a typical genome. For example, the coronavirus is made of RNA, and its genome is 26 to 32 kilobases. That means it's about 30,000 bases, whereas a cell's genome is approximately 10 million bases. So they're much, much smaller. The DNA of the virus directs the host cell. So it takes over the host cell and gets the host cell to make new virus copies for itself. Now, if it's an RNA virus, then it can be directly transcribed. It doesn't have to worry about the host cell's DNA or replicated first. So RNA viruses use enzymes that can make more errors, and the RNA viruses mutate more frequently. So these are some components of RNA viruses that can cause more problems for us. So how do we classify viruses? Well, this is not a course in virology. So we're just going to take a look at a few of the ways. So we can either classify viruses based on their type of nucleic acid, DNA or RNA, and its function. We can classify them based on the capsid structure, the protein outer structure. We can look to see whether they're enveloped or non-enveloped, or we can look at the actual genome structure, how the genome is put together. We're going to really look at only the first two. So here we see that we have some DNA viruses, and it gives you a couple examples. I'm actually not going to require you to remember these, but just know that these are some viruses you may have heard of. So the rabies virus is made of DNA. Herpes virus is an RNA virus. These tell you who are single-stranded, who are double-stranded. So you notice that the rabies virus is a single-stranded DNA, which is different from what more complex what cells have. And the herpes virus, which is RNA, is double-stranded. And then it goes on to talk about whether this DNA is linear, circular, broken in pieces, or one continuous piece. Again, you don't need to know these examples. Another way that we can classify viruses is based on their capsid structure. Remember, that's the protein that is around the outside. So we have the helical form, which is a spiral shape. And we have the icosahedral form, which for the icosahedral form, we are talking about a capsid, a coat that has 20 sides made up of equilateral triangles. So you notice in here that it could be a naked icosahedral, it could be an enveloped, so it might have an envelope, an additional coat on the outside. You could have an enveloped helical, or a naked. By naked, it just means it doesn't have the extra envelope on the outside of them. And here we see that this is more based on the genome structure. You do not need to really know anything about this. This is the Baltimore classification that looks at whether it's double-stranded, single-stranded, etc. But that's just another way to classify viruses. Here's a video link that you can take a look at on your when you have a moment to look at how a virus, an animation of how a virus gets inside of a cell. So once a virus does get inside of a cell and takes over the host machinery, it can cause some damages when it is making additional viral particles. Some of the potential damages that it can cause the host cell to lyse or break apart, and that would obviously kill the cell. It can also cause the cell to go through apoptosis. This is when the cell realizes that something is not right and it goes into a self-destruction mode. So the cell actually kills itself because it recognizes that something's not right, but you still end up with dead cells. And even if it doesn't kill the cell, you can end up with widespread symptoms on the body. So you know when you get the flu, it's not just that you might lose some respiratory lining cells, you are also going to feel really awful. Oh, so if in fact the virus doesn't kill the host, what it can do, it can leave the host by budding, which means that it sort of breaks off an exocytosis, which doesn't actually in fact kill the cell. So when a virus is about to infect its host cell, there are 4 stages. First, it must attach to the outside of the cell. Then it's going to enter the host cell, and it does it in a variety of ways. Then it needs to go through its replication and assembly of new virions, and then those virions need to be released or let out of the host cell. So let's go through attachment. So attachment, there are receptors on the surface of the host cell that the virus is able to bind to. So it recognizes the receptors on the host cell and is able to bind to that cell. The virus can attach only to cells that have the right receptor molecules. For example, the coronavirus, the COVID-19, attaches to the ACE2 receptors on cells, on host cells. Therefore, viruses can be very specific about what cell or even what species they're infecting. So here, once they attach, we're going to have an entry point. So they can enter in a number of different ways. They can just merely inject their genetic material, be that DNA or RNA, or they may actually be taken up by the cell. So here we have a bacteriophage, which lands and attaches to its bacterial cell, and then it literally injects only its DNA. The capsid stays outside. Whereas for some viruses that attack eukaryotic cells, they may attach and literally be taken up by the cell through endocytosis, or they may just attach and inject their genetic material. So once they're inside, they have to take over the cell machinery and begin their assembly of new virions. So if they are a DNA virus, then they can use the machinery to transcribe new messenger RNA, which will make viral proteins, and they can duplicate or copy using DNA polymerases and make new viral genomes. And then those can be assembled into new virions. If they're an RNA, a regular RNA virus, they can make a complementary RNA if that's required, and then they can transcribe. Some RNAs can be directly transcribed into messenger RNA to make viral proteins, and then they can copy their RNA to make new viral genomes. They can use an RNA polymerase to make new viral genomes. If they are an RNA retrovirus like HIV, what travels with them is their own enzymes to reverse transcribe the RNA into DNA. They use a reverse transcriptase that they carry with them. So they transcribe their own RNA into DNA. That DNA can then get into the host genome, and then we would follow the same path as what was up here, transcribing into messenger RNA to make new viral proteins and duplicating the DNA to make new viral genomes. I'm sorry, in this case, they would be copying and making messenger RNA to make new viral genomes. Then when they need to be released, remember that this can involve lysis, where the cell, here's our host cell, literally is burst open and the viral particles exit looking for new hosts, and the host cell dies. Whereas additionally, what can happen is called budding. So here you have a virion that needs to be released, and it can exocytose and be released, and that does not directly kill the host cell. So here are the steps for infection, and this particularly is an influenza virus. So it is attacking a eukaryotic cell, an epithelial cell. Here's the nucleus of that epithelial cell. The virus binds. It is endocytosed. It's taken inside the cell. It's then broken apart, and the viral RNA enters into the nucleus. It gets replicated by a viral RNA polymerase. That viral RNA polymerase is used to make the viral messenger RNA that is made is then used to make viral proteins. Those viral particles are then made and incorporated into the virion, and then the cell buds off. The virion buds off and is released. So it doesn't kill the cell, which can continue to make new virus particles. Now, here's an electron micrograph of those bacteriophages. Here you see one onto a bacterial cell. And in bacteriophages, we see, or in the infection of bacterial cells, we see there are two stages. One stage is called the lytic cycle, and one is called the lysogenic cycle. So in the lytic cycle, we have what we just went over. The bacteriophage binds. The genetic material comes into the cell. This DNA forms. This is the viral DNA. This is the host DNA. We're going to use this to make more. We're going to replicate this DNA, make more of this DNA, and then make more proteins for the capsid, reassemble, and then we're going to see that the cell lyses. Additionally, we can have a lysogenic cycle happening at the same time, where the phage attacks, just like we saw above, and the DNA, instead of just making a circle, so this can happen rather quickly, it can incorporate itself into the host DNA and hang out there, get passed on through subsequent divisions of this bacterial cell, and that at a later stage can actually release the viral DNA, which can replicate, and we can make more copies of the DNA, and then it can enter in at this point and eventually cause the release. So having these two separate stages is beneficial to the virus because it can hang out in a population of bacterial cells. Now let's take a look at some of the effects of animal viruses. And so animal viruses may cause an acute disease. That means acute disease is happening right now. It could cause a chronic infection, something that's ongoing. So the virus is maintained in the body, and hepatitis C is an example of that. Could cause latency and intermittent symptoms, meaning that the organism will have the virus in its body for its lifetime. And there are times when the virus is not active, and then times when it becomes reactivated and then starts going through an active phase of replication and causing the symptoms of the infection. And so an example of that is herpes simplex. We could have asymptomatic infection. So there's other herpes viruses where you don't actually know that you are infected. And it could cause oncogenesis, which is the formation of a cancer or the transformation of the host cells into cancer cells. So hepatitis B and C and the human papillomavirus are capable of doing this. Hepatitis B and C causing liver cancer, and human papillomavirus causing cervical cancer. Now let's take a look at some plant viruses. Now remember, viruses can attack all kinds of living things. So there's horizontal transmission. So think horizontal is on a straight line. So this is when a virus enters a plant because of a damaged cell tissue. So it's able to break through the plant's defenses. So and then it might be transferred from one plant to the next via things like pollen. So here we see one plant, if it becomes infected, it can pass that virus to a neighbor plant. So that's horizontal transmission. And then we have vertical transmission, where the virus is transferred from a parent to its offspring. So here we have vertical transmission to a new offspring plant. In both cases, either transmission can cause decreased growth and vigor of the plants and may even cause death of the plant or the plant's tissue. So impacts on humans. You don't need to know any of this. It's just to show you the variety, and you may have heard of some of these different viruses and where their impact is on the body. Now, luckily, we have vaccines which can help prevent viral infections. So how vaccines work is they trigger immune protection. They teach your body how to recognize viruses when they infect and to remove them before they become dangerous or cause damaging effects. So how can we prepare viruses? We can either take, now here we call this a live virus. Again, this is a problem for me because they're not live, but what this means is a completely intact virus, and they can attenuate, so they can break a piece of it. And that way they can use that to teach your body. So there's very small risk of infection. They can also use a killed virus, which means that it's a really broken virus. There's no way that this can, when it's injected into your body, that this can give you the disease. So that's what the flu vaccine is. And then lastly, they can use molecular subunits, and that's what the COVID vaccine is. We can use bits of messenger RNA that code for the little component spikes on the coronavirus. And so that teaches your body to recognize those little spikes. If you do become infected, there are a number of antiviral drugs that we've developed. Now, these don't make the virus go away, but they help. So what they do is they can reduce the number and the duration of a viral episode. For example, there's a drug called acyclovir that helps to do this, reduce the number of viral particles for herpes. And if there is an outbreak, then it shortens the time of that outbreak. There are some antiviral drugs which reduce symptoms like Tamiflu. So Tamiflu is obviously for influenza. So it turns off an enzyme that the virions use to get out of their host cells. There are antiviral drugs, and you'll recognize this name, remdesivir, which introduce faulty subunits. So they introduce nucleotides that are wrong into the viral RNA polymerase, and viruses need that in order to make new RNA. So this is one that has been used in COVID treatments. And lastly, they can actually block the virus's replication, the making of the new virus. So AZT is a drug that's used to treat HIV, which inhibits reverse transcriptase. Remember, this is a retrovirus, so we need the reverse transcriptase to turn the RNA into DNA. And if we block that, then that will really, really decrease the number of HIV particles in an HIV positive person. It doesn't completely rid that person of the HIV infection, but it makes it so their viral load is so low it's undetectable. And again, don't need to know the details of this diagram because there's many. A couple things, interesting things to notice. HIV is an enveloped, so it has a coating. Icosahedral, so it's one of those equilateral triangle unit viruses. It's capsid. So when it attaches to its host cell, its white blood cell, T cell, it injects in its RNA, and it needs a reverse transcriptase to turn that around, but yet the AZT will block at this point, and so we cut it off right there. Can't go through the rest of the process. The last two slides have to do with particles which are not technically viruses, but they're other small infectious particles. So we have things that are called prions. Now these are bits of protein that are infectious. They're very, very small. They have no DNA or RNA, so they have no nucleic acids. And there are forms that are they're proteins that are not folded correctly. So the body doesn't recognize them, and they insert themselves and cause a whole scheme of fatal neurodegenerative diseases. So you may have heard of these: mad cow disease, the Creutzfeldt-Jakob disease in humans, Kuru is another one, scrappy in sheep, chronic wasting disease in deer. These little misfolded proteins get inserted into nervous tissue and cause incredible damage. And unfortunately, there is no treatment. And unfortunately, prions are not destroyed by cooking. So if you eat products from animals that are infected, they could infect the human. And then our last slide is about viroids. So these are don't get that confused with virions. These are very small circles of RNA only, no capsid, no envelope. They're only known to infect plants They do get into their plant host cells and replicate within them. They don't make any proteins because all they are is a bit of RNA that doesn't code for any protein structures, and they can cause devastating crop failures. All right, thank you.